topic is General Mycology and Overview of Fungal Infections. Mycology is the branch of medical science that deals with the study of medically important fungi. The name fungus is derived from Greek myces, meaning mushroom, a type of edible fungus. Now, fungus differs from the bacteria and other eukaryotes in many ways. Fungi are the eukaryotic and they possess all the eukaryotic cell organelles such as mitochondria. They possess a rigid cell wall composed of chitin, beta-glucans, and other polysaccharides. Fungal cell membrane contains ergosterol instead of cholesterol. Fungi may be both unicellular or multicellular. Fungi generally lack chlorophyll and divide by asexual or sexual means by producing spores. The morphological classification of fungi. Based on the morphological appearance, there are four main groups of fungi which are shown in this table. First is the yeast. They grow as round to oval cells that reproduce by mainly asexual process called budding, in which cells form protuberances which enlarge and eventually separate from the parent cells. The examples are Cryptococcus neoformans, which is a pathogenic yeast, and Saccharomyces cerevisiae, which is a non-pathogenic yeast. Next group is the yeast-like. In some yeast, for example, Candida, the bud remains attached to the mother cell, elongates and undergoes repeated budding to form chains of elongated cells known as pseudohyphae. They can be differentiated from the true hyphae as they have constrictions at the septa. So these yeast-like, they form pseudohyphae. Next is the molds or filamentous fungi. The examples of these are the Aspergillus, penicillium, and dermatophytes. They grow as long branching filaments of 2 to 10 micrometer width called hyphae. The hyphae are either septate, that is, they form transverse walls, or non-septate. There are no transverse walls and they are multinucleated, that is, coenocytic. The hyphae grow continuously and form a branching tangled mass of growth which is called mycelium. So the mycelium, based on the mycelium, it can be divided into aerial mycelium and vegetative mycelium. So the examples of molds are Aspergillus, dermatophytes, penicillium, rhizopus, mucor. Next is the dimorphic fungi. They exist as both molds in the environment at ambient temperature, that is 25 degrees Celsius, and as yeast in human tissues at body temperature at 37 degrees Celsius. Some medically important which are dimorphic, such as the Histoplasma capsulatum, the Blastomyces dermatitidis, Coccidioides immitis, Paracoccidioides brasiliensis, Penicillium manefi, and Sporothrix schenki. So these are the morphological forms of the fungi. First is the budding yeast, as you can see. Next is this is the budding yeast cell. Next is the budding yeast with pseudohyphae. Example is Candida. This is the aseptate hyphae, which is molds for filamentous fungi, and this is a septate hyphae. You can see here septate formation. This is, for example, Aspergillus, and these are the mycelium. So the dimorphic fungi will both have this yeast form and the septate form. Next is the taxonomical classification. Based on the production of sexual spores, the kingdom fungi can be divided into four medically important phyla. That is, first is the phylum Zygomycota, next is the phylum Ascomycota, third is the phylum Basidiomycota, fourth is the phylum Deuteromycota. So phylum Zygomycota, they are lower fungi and they produce sexual spores known as zygospores and possess aseptate hyphae. For example, rhizopus and mucor. In phylum Ascomycota, they produce sexual pores known as ascospores and possess septate hyphae. Example, Aspergillus. In phylum Basidiomycota, they produce sexual spores known as basidiospores. For example, Cryptococcus. In phylum Deuteromycota, that is phylum Fungi imperfecti, in majority of the medically important fungi, the sexual state is either absent or unidentified. Hence, they are traditionally grouped as fungi imperfecti. So these are the types of spores which are produced. That is, the sexual spores produced by zygomycetes is zygospores, by Aspergillus is ascospores, by Cryptococcus is basidiospores, and vegetative asexual spores which are observed are arthrospores in Coccidioides and Trichosporon, blastospore in Candida, chlamydospore in Candida albicans. Some aerial asexual spores are conidospores or conidia in Aspergillus, sporangiospore in Zygomycetes, microconidia in dermatophytes, macroconidia in dermatophytes. Next, we move on to the lab diagnosis of fungal disease. So the laboratory diagnosis of fungal disease mainly comprises of first we begin with the specimen collection. It depends on the site of fungi infection such as fungal infection such as skin scraping, nail, hair, sputum, etc. For systematic mycosis, we need to uh, take blood sample, cerebrospinal fluid, which is for collected for cryptococcal meningitis. So this you can see, this is the how the nail clipping can be taken. This is the skin scraping, and this is how the blood cultures are taken. Next, first we'll do microscopy. So the fungal elements can be detected in the clinical sample by direct microscopic examination of the material from the lesion. So the first we can use is potassium hydroxide preparation. The keratinized tissue specimens such as skin scrapings and plucked hair samples are treated with 10% KOH, which will digest the keratin material so that the fungal hyphae will be clearly seen under the microscope. So for about 10% of the usual concentration of KOH will be used normally. About 20 to 40% KOH will be needed if we need to for hair and nail for digesting the sample. So this is how it looks in the KOH preparation of the fungal hyphae. How the fungal hyphae will look clearly. For the biopsy specimens, it needs to be dissolved in 10% of KOH in a test tube and examined after overnight incubation. Glycerol 10% can be added to prevent drying of this KOH preparation, and dimethyl sulfoxide can be also be added to, which will help in more to digestion. Next is the gram staining. It is useful in identifying the yeast, example Cryptococcus, and yeast-like fungi, example Candida. They appear as gram-positive budding yeast cells. The ink, we can also use negative stains for demonstration of capsule of Cryptococcus neoformans. Calcofluor whites, it has more sensitive than other stains. It binds to the cellulose and the chitin of the fungal cell wall and fluorescence under UV light. Ultraviolet. Histopathological stains also can be used for demonstrating fungal elements from the biopsy tissues, that is periodic acid shift stain. Pass positive fungi will appear magenta or deep pink, whereas nuclei will appear blue, and periodic acid shift stain which stains only the light stain, the dead fungi. Next, histopathological stain can be used as Mason Fontana stain for pigmented fungi or pheoid fungi. Hematoxylin eosin stain can also be used for tissue samples, and mucicamine stain for staining the carminophilic cell wall of the Cryptococcus and the Rhinosporidium. Another very important stain for histopathological stain is the gomodimethamine silver stain. It is an alternative to the PAS stain. It stains both live and dead fungi and stains the polysaccharide component of the cell wall. The fungi will appear black and background will appear pale green color. Next stain which can be used in microbiology for fungal elements is the lactophenol cotton blue stain. It is used to study the microscopic appearance of the fungal isolates grown in culture. It contains phenol, which acts as a disinfectant, lactic acid, which preserves the morphology of the fungi, glycerol, which prevents the drying, and the cotton blue stains the fungal elements blue. I showed you an SDA plate which is having growth of Aspergillus. Plant is having a growth of Aspergillus. The cornmeal agar and the rice starch agar, it is a nutritionally deficient media which is used for the stimulation of chlamydospore production. Chlamydospore production will only take place only when there is spore production will be taken when there is nutritional deficient. So we use such medias to see the what pattern of spore will be produced. Mostly chlamydospore will be produced by Candida. Next is the brain heart infusion agar and the blood agar. This enriched media will be used for growing the fastidious fungi like Cryptococcus and Niger seed and bud seed agar. It is used as a selective media for growth of Cryptococcus. Chrome agar Candida media. It is used as a selective as well as differential media for the speciation of Candida. So you can see here Candida tropicalis. By the differentiation of the colors, we can differentiate the species of different Candida albicans along with the non-Candida albicans. For the culture to grow, we need a specific culture condition. So these are the following culture condition which is required for the growth of the fungi. Temperature, most of the fungi will grow well at 25 to 30 degrees Celsius except the dimorphic fungi that will grow at a temperature at both 25 degrees Celsius and 37 degrees Celsius. So we use biological oxygen demand incubators which help in the growth of fungus. The incubation time for the rapid growers will be from 4 to 5 days, and for slow growers it will take around 2 to 3 weeks time for the fungus. So minimum for a fungus we should keep in the incubator for minimum of 3 weeks time before identification. Next is the antibiotics. For so like me if media can promote the growth of antibiotics, so we use certain antibiotics like cyclohexamide and chloramphenicol and gentamicin to inhibit the growth of bacteria as well as some saprophytic fungi, so that we can appreciate the pathogenic fungi. For correct identification of the fungus, it is based on the macroscopic appearance of the colonies grown on the culture and microscopic plate based on the LPCB mount of the the macroscopic appearance of the colony will be depending on the rate of growth, that is rapid growth if it less than 5 days, which is mostly seen in saprophytes, yeast and agents of opportunistic mycosis. Then slow growths will be seen in 1 to 4 weeks, which is observed mainly in dermatophytes and agents of subcutaneous and pigmentation, which will be seen on the reverse side of the culture media. Texture, it refers to how the colony would have felt if allowed to touch. It may be of various types such as glabrous, velvety, yeast-like, cottony or granular or powdery. Then we check out for the microscopic appearance of the fungi, we need to do T's mount. Identification will be based on the nature of the hyphae, that is septate or aseptate, hyaline or furred, narrow or wide, and type of sporulation, that is conidia or sporangia. Slide culture, we do in situ microscopic appearance of the fungi. So though this is a tedious process, that is slide culture, but it gives the most accurate in situ microscopic appearance of the fungal colony. A sterile slide is placed on a bent glass rod in a sterile Peptide dish. Two square agar blocks measuring around 1 centimeter square, which will be smaller than a cover slip, are placed on the slide. The bit of, you can see here, the two small squares, that is 1 centimeter squares area, are placed on the slide. Then the bit of fungal colony is inoculated at the margins, onto the margins at the center of the agar block. So it will be inoculated in the margins. Then the cover slip is placed on the agar block and the petri dish is incubated at 25 degrees Celsius. After the sufficient growth occurs, the LPCB mounts will be made both from the cover slips and both from these cover slips and the underneath slide. So this is the method of slide culture for microscopic appearance of the. Next method we use is a cellophane tape method. In this, the impressions are taken by placing the cellophane tape on the colonies present on the surface of the subarouse dextrose plate, agar plate. Then LPCB mounts will be made from the cellophane tape. This is a easier method as compared to slide culture and helps in situ, and it will maintain the in situ fungal morphology. Okay. There are some other methods for identification of various different fungi. That is, for Candida, we can use germ tube test, Dalmau plate culture, carbohydrate fermentation test, and carbohydrate assimilation test are done. For dermatophytes, hair perforation test can be done. Dermatophyte test media can be used, and dermatophyte identification media can be used. For urease test can be done for fungi that produce urease enzyme. For example, Cryptococcus. Here, urease test will be rapid urease. Certain immunological methods can also be used for identification of fungus, and hence it will help in lab diagnosis. So first is the antibody detection by ELISA immunodiffusion method, agglutination test and complement fixation test. Antigen detection by latex agglutination test. Various fungal antigens can be detected in the clinical samples, such as blood, cerebrospinal fluid, urine, such as, for example, cryptococcal capsular antigen formed CSF by latex agglutination test can be done. Detection of Aspergillus specific galactomannan antigen in patient sera or urine by ELISA can be done. Beta D glucan assay will help in detection of beta D glucan antigen in blood by ELISA. It is a marker of invasive fungal infections, which is raised in all invasive fungal infections except for zygomyces, blastomyces and cryptococcus. Some immunohistochemistry methods can be used. Actually, it refers to the detecting antigens on the cells of a tissue section by using fluorescent tagged antibodies that bind specifically to the antigen. It also helps in diagnosis of deep mycosis. Automated identification systems such as Marlitov and Vitek, they are revolutionary in accurate identification of yeast and to some extent molecular methods. We can use polymerase chain reaction and its modification, that is multiplex PCR, nested PCR, and the most advanced real-time PCR. And we can use some DNA sequencing methods which have been developed for accurate identification of the fungi from culture as well as from the specimens.
Thank you for watching this video. If you enjoyed it, please hit the like button and the bell notification icon.